The overall goal of the following experiment in a genetically trackable metazoan Caenorhabditis elegans is to identify tissue-specific chaperone interactions.
The main advantage of this technique is that it combines easy-to-use assays, such as feeding RNAi and behavioral readouts, to expose novel tissue-specific interactions.
For a non-stressful embryo synchronization, use a worm pick to move about 100 eggs from an unsynchronized worm plate to a newly seeded NGM plate and cultivate the animals according to the appropriate experimental schedule. When the animals reach the first day of egg laying, slowly add 1 milliliter of M9 buffer to the plate distant to the bacterial lawn and rotate the plate so that the buffer completely covers the plate. Tilt the plate to one side to remove the liquid and wash the animals from the plate. When all of the worms have been washed from the plate, use a standard plastic pipette tip to cut a square of agar around where the eggs are concentrated and place the piece of agar onto a newly seeded NGM plate. Cultivate the eggs under the same culture conditions. At the end of the incubation, the plate should be covered with synchronized eggs. After washing all of the animals from the plate as just demonstrated, add 1 milliliter of fresh M9 buffer and use a cell scraper to release the eggs from the plate. When all of the eggs have been detached, transfer the entire volume of buffer from the plate into a conical tube for centrifugation. Centrifuge the tube and discard the supernatant. After removing the supernatant, add up to 1 milliliter of fresh M9 buffer to the plate and pipette to resuspend the eggs. Wash the eggs 5 more times as just demonstrated. After the last wash, the egg pellet should appear white.